In this lab video, we're going to go over the mixed ANOVA and how to perform those analyses using Jamovi and other various follow-up calculations that won't really use Jamovi for. We'll do them by hand. Um, the data, the example for this lab comes from a paper that I published recently. So this is a paper that I published in Journal of Business and Psychology with some colleagues looking at how people respond to velocity, changes in their velocity as they perform a task. So velocity is the rate of progress that a person makes while they perform their task. And so there's some literature showing that people uh, generally don't like going slower than they want or expect to, that they find that to be frustrating. Uh, but there's some confounds in that literature, and that's one of the purposes of this paper is to sort of address those confounds. Um, So what I'm going to do here is I'm going to just grab text right from the manuscript, and we have the data from the paper. And what we're going to do is we're going to try to recreate what is in the manuscript, following along with our data. So you see here that we started with 95 undergraduate participants, but three of them had technical difficulties. So actually, our final sample size is 92. So that means we have 92 participants in this study. But recall that it's a mixed design, mixed ANOVA. And so we have 92 people, but each person will be giving us repeated measurements, and each person will be assigned to different between subjects conditions. So we look at the procedure, it gives us some information about this. You say, okay, so the study was online, participants signed on, they gave consent. They were given detailed instructions for how to complete the task. Following these instructions, participants recorded the degree to which they were frustrated at the moment. So basically, what this is saying is before really doing anything, participants were asked, how frustrated are you right now? And this served as the baseline measurement of frustration. Then after this, participants started performing the task. So they completed a practice trial and they got to experience the experimental task. And then they were assigned to the velocity referent manipulation. So it doesn't say it here because there's another section of the paper that describes it. But just briefly, uh, half of the participants were given, they performed the experimental trial under conditions that were kind of slow, except they didn't know that those conditions were slow. So then when they went and performed the actual trial, that was just normal to them. The speed didn't change, it was all fine. The other half of the participants performed the practice trials under conditions that were kind of fast. They were able to get through the task pretty quickly. Then when they got to the actual trials, everything slowed down for them. So these folks were going slower than they were accustomed to. So you had two conditions. One group of people who were going at the speed that they expected and thought was normal, and another group of people who were going at a speed that was slower than they thought was normal. In actuality, though, everyone was going the same speed. So after we manipulate velocity, we measure frustration again. So this is immediately after it's manipulated, but before they begin the actual trial, the actual task that they're going to perform. So they know that their velocity has been changed, but they haven't performed the task yet. So this is the second measurement of frustration. Then they perform the task. Uh, this task was important to them because they could earn money by performing it. The better they did and the more they got done. And immediately after they completed that task, frustration was measured for the third time. So this is the post-trial measure of frustration. Then after that, there were some other measures and participants were debriefed. But basically, the key issue here is that we had a measurement of frustration before anything happened. Then velocity was manipulated. Half the people were in the slow condition and half the people were in kind of the just normal condition. Then immediately after that, frustration is measured again. Then they do an experimental trial and then it's measured again. So we have 3 measurements of frustration. If we look at our data, it looks like this, right? If we can plot the means. And so I've went and grabbed these means from Jamovi. And why don't we go ahead and see how to do that? So let's pop over to Jamovi and open up our data file. We have to remember, navigate to where we've stored it, open it up. And we have our velocity dummy condition. This is just which condition they're in, 0 or 1. And our 3 measurements of frustration, baseline, pre, and post. Well, I wanted those means, so I'm going to go over to exploration and descriptives and say, well, I want the mean for baseline, pre, and post, so I'm going to move those over. And I want to split that by condition. I want to know what the means are in each condition. There's a lot going on in this table. Really, I just care about the means right now. So that's all I'm going to ask for from that table. And that's what I have in the slides, just the mean, depending on which condition you're in, and the at each measurement period, baseline, pre, and post. So if we look here, we have these means plotted. So if we look in the below referent condition, which is just coded 0 in the data, we can see we have a mean at baseline is 1.39, and that's plotted there. The mean at pre-trial in this condition was 1.78, that's plotted there, and the mean at post-trial was 1.73. Likewise, in the control condition, the mean at baseline was 1.46. The mean at the pre-trial was 1.41, and the mean at the post-trial was 1.36. So we have a first things first, we like to plot our means, get the picture, look and see what's happening. At a glance, it looks like frustration is changing over time on average. It also looks like frustration is different across condition on average. So those would both be main effects. But it also looks like there's an interaction. It looks like there's definitely a different effect of time on frustration depending on which condition you're in. But of course, to know this, we have to go forward and run those statistical tests. So pulling from the paper, we have here where I write about this and says, okay, hypothesis 1 stated that the below referent velocity would be associated with increased frustration. So we began testing this hypothesis, with momentary frustration. We computed a 3x2 mixed analysis of variance. So our within subjects factor is time, and that had 3 levels: baseline, pre-trial, and post-trial. Our between subjects factor is condition, and that had 2 levels: below referent and control. It's a mixed ANOVA, which means that it's a within subjects ANOVA. It has a within subjects component. So if we look at this footnote here, footnote 7 says, well, okay, first things first, we want to look at Mauchly's test to see if we have sphericity assumption violations. And we don't. So we can just sort of move forward, assuming compound sphericity. So then moving forward, I said, hey, looking at this figure, it looks like the below referent group has higher frustration than the control group on average, but actually that wasn't borne out through the statistical test here. We see that there was no significant main effect of the velocity condition on momentary frustration. You have an F degrees of freedom, mean squared error, a p-value, and it's, p-value is greater than 0.05. That's not significant. But what's actually important here is that there is an interaction. So we see there's a time by condition interaction. Again, an F with degrees of freedom, mean squared error, p-values, and that's statistically significant. We have a note here, see figure 2, and you can see in figure 2 that we've been looking at all along that, yeah, this indeed looks like an interaction. It looks like the effect of one condition or one factor rather on the dependent variable depends on the level of the other factor. It looks like the effect of time on momentary frustration depends on the level of condition, which condition you're in. So let's go and see how we can compute this and see if we can compute this in Jamovi. So here I have the output. Let's go over there and see if we can generate this and replicate what is in the manuscript here. So if I come over, I'm going to be doing an ANOVA, and it's a repeated measures ANOVA. So I need to set up my repeated measures factor. We're just going to call it time. It has 3 levels, baseline, pre, and post. And so I'm going to go ahead and move these over to the appropriate spot. I move baseline over to baseline, pre over to pre, post over to post. But it's a repeated measures ANOVA, but it's actually a mixed ANOVA, right? Because I have both within subjects factors, time, and a between subjects factor. So I want to go ahead and move over this velocity between subjects factor. And we can see here that we get this both a within subjects table and a between subjects table. And really the within subjects table is where it's going to tell us about this interaction. And we can see that we do indeed have a time by velocity dummy interaction. And if we go over and look at the slides, it's a little bigger. We can see that the values reported here. So for this between subjects main effect, I had an F of with 1 in 90 degrees of freedom. So 1 in 90 over here has an F of 2.12. The mean squared error was 1.6, and that was not significant at p-value of 0.149. But the interaction term had an F of 5.15 with 2 in 180 degrees of freedom, a mean squared error of 0.28 with a p of 0.007, so that is statistically significant. Notice I didn't report in text the main effect of time. It's non-significant. It's not that important because the main effect doesn't really matter that much because, or in this case, the lack of a main effect doesn't really matter that much because we have this interaction. So recall, as we talked about in class, if you have this interaction, it tells you that the main effect isn't really telling you the whole story. So you really need to dig deeper. Oh, and one last thing. Yeah, Mauchly's test is reported down here. I had reported a chi-square because I did my analyses in a different software called SAS, where they report chi-square. Jamovi is not reporting that statistic, but it's fine. You can see the p-value is exactly the same. That's really what we care about. We just know that our assumption is not violated, so we're going to move ahead and not worry about adjusting for the greenhouse geyser. So we have a significant interaction. We want to then go and probe that interaction and do our follow-up probing tests. So in the paper, we begin by doing between subjects tests. So we say, well, as you would expect, between subjects follow-up tests were, there was no difference between the two conditions in frustration during the baseline measurement period. And you'd expect that because at that point, the two conditions actually weren't different. This was the baseline measurement. This was as people showed up to the experiment. There really shouldn't have been a difference across condition. So we didn't expect one. So you say, okay, well, you have a T with 155 degrees of freedom is negative 3.39, a standard error of 0.18, and a p-value of 0.7. How do we get these numbers? Well, it's a t-test, right? We're comparing two means, but it's a between subjects t-test in the context of a mixed ANOVA, which means we're going to have to go and compute those mean squared error and degrees of freedom. So first of all, let's look at and be clear about what we're assessing. We're assessing the difference between these two means. We're looking at the simple effect of condition below referent versus control at baseline. So this is a simple effect. We're comparing these two means. We're going to do it with a t-test with some degrees of freedom. We know that the numerator is the difference between these two means, but we need an estimate of our mean squared error, and we also need to know the sample sizes for these two conditions. So for the sample size for the 0 condition, that's the below referent, and the sample size for the 1 condition, that's the control group. So let's go and grab those sample sizes first. You can see I put the descriptives here. I did this the same way I got the means before. If we go over to Jamovi and I go to explore and I go to descriptives and I move over my variables, well, let's put them in order. Yeah, okay, hold on, bear with me. There we go. Baseline, pre, post. And I want to split them by velocity condition. And in this case, I just want the n. So I'm going to unclick all these boxes. I just want the sample sizes. And I can see here that in the below referent condition, I had 45 people. And it's the same at every time period because I have the same number of people at each time period. No one dropped out of the study. And I had 47 people in the control condition. So if we come back to the slides, that's what we have here. So in my equation, I can pop that 45 and 47 into the equation there. Then I still need to find the mean squared error and the degrees of freedom. And of course, I do that with my hybrid equations here that we've discussed in class, where I'm dealing with a mixed ANOVA and I'm dealing with between subjects simple effects. I need to compute this sort of hybrid mean squared error and hybrid degrees of freedom using these equations. As I've recommended in class, I made myself a calculator years and years ago in Excel. So I don't calculate this formula anymore. I just type it into Excel and it comes out perfect every time. So we see that I have a mean squared error of 0.722 and a degrees of freedom of 155. So I can go ahead and pop that in to my equation there. And then it's just a matter of solving, where I have in the numerator, the difference between those means was negative 0.07. My standard error, which is my denominator, is 0.18, and that comes out to a t of negative 0.39. Just a quick note about the calculations on the slides here. They might not match exactly, like there might be rounding error, because when in the paper, I did this rounding out to like however many decimal places SAS rounds out to 8 or 10 or maybe even more. Whereas for the slides here, I've just rounded to 2 decimal places. So if you divide those and you go, hey, it's not exactly 0.39, that's why. It's just rounding error. So then you go, okay, well, conversely, during the pre-trial and the post-trial measurement periods, participants in the below referent velocity condition did indeed experience higher frustration relative to participants in the control condition. So we look at the pre-trial. Now we're doing the simple effect of condition at pre. And here the difference between the two means is 0.36. Our standard error is the same as it was before because all of that information was drawn from the omnibus test, right? So it's just 0.18. And we can divide that. And this is an example. 0.36 divided by 0.18 is actually 2.0 exactly. But because of rounding error, the t is actually 2.05. And we can do the same thing here. The simple effect of condition at post-trial is just this t with 155 degrees of freedom. Again, the standard error is 0.18. And the difference in those means is 0.37. So that comes out to a t of 2.1. Then in the paper, we move forward to do within subjects test probing. So the first thing you might say is, well, okay, in the control condition, is there a significant effect of time in the control condition? And what we find here is, no, there's not. There's no significant effect of time in the control condition. Or said differently, momentary frustration did not change across these 3 measurement periods. Any difference in these means is just sampling error. It's not to be interpreted. So let's talk about how we got this answer. So we want to go back to Jamovi, and now we want to split our file and look at just the control condition. Okay, we want to look at just the control condition and look at this ANOVA. To do that, recall that we have to go to our data and we have to create a filter where we have a filter where our velocity condition, so valcon, that's the, or valdum, excuse me, equals 1. We just want that. So you can see anytime it equals 0, it gets X'd out. Those people are now not in the data. They're not included in anything I do. So if I come back over here to analyses and I rerun my one-way ANOVA where I have my factor is time, I have baseline, pre, and post, and I move over those variables, baseline, pre, and post. I'm not splitting by velocity condition anymore. I'm not doing that because I'm only looking at one velocity condition. I'm only looking at the control condition. And I come here and I can see it says, okay, this F is 0.58 and it's not significant. But hopefully some alarm bells are going off here. I only have 92 degrees of freedom. I've split my data in half. I've only looked at half of my data. And so when I'm estimating my mean squared error here, my estimate of population variance, it's not as good of an estimate as I could. I should be using all of my data to do this. So let's go ahead and grab this table. And I've pasted it there. This is that same table we're just looking at. But remember, I just want to use the sums of squares for the time variable. It says RM factor 1. It's time for that time variable, right? So for the error term, I want to grab the error term out of my omnibus results. So I'm going to replace what's there with what's in my omnibus test results. So now my F for my mean squared error is going to be 0.108 divided by 0.283, which is 0.38, which has a p-value of 0.683. Now, in this case, it didn't change any interpretation, but it could. And so this is what we want to do. We want to use this better estimate of mean squared error from the omnibus test. Now, in the previous in the slides, I was doing the same thing, but I had to compute that hybrid mean squared error because I was doing between subjects effects. Here I'm doing within subject simple effects, and so it's much simpler. I just have to grab that mean squared error right out of the omnibus test, out of the omnibus table, put it here, and recompute my F. So if I come back to what's reported in text, we see that, yes, I have an F with 2 and 180 degrees of freedom. I'm using all of my data. My mean squared error is 0.28, which is the same as it was from the omnibus test. And that has a p-value of 0.683. Moving forward, I say, well, okay, but looking at the below reference condition, there was a significant change in frustration, across time. There was a significant change across time. But now, okay, there's a change. We have three means here: baseline, pretrial, and post-trial, 1.39, 1.78, and 1.73. We know that at least two of these means are different, but we don't know which ones. We need to do further follow-up tests now. So, just how I got this again, same way. I went to Jamovi, except this time I go to my data, and I'm going to change this filter to do velocity condition equals 0. And so now you can see it's only keeping the people in the 0 condition. And I come down here, and it has updated my repeated measures ANOVA with an F of 5.34. But recall, it's only giving me 88 degrees of freedom. My mean squared error is not what I want it to be. So I go here and I replace that with the mean squared error and degrees of freedom from my omnibus test, recompute the F, and that is the value that's reported in the manuscript. Okay, so again, what I was saying is we know that at least two of these means are different, but we don't know which two are different. So we're going to follow up with some t-tests. So first, relative to the baseline measurement period, momentary frustration was higher during the pre-trial. So we're comparing baseline to pre-trial within the below reference condition. So the numerator of this t-test is 1.78 minus 1.39. The denominator, I have my mean squared error is 0.28 because that's just right from the omnibus test. And the ends are both 45 because you have 45 people in this condition. So there were 45 people during the baseline and 45 people during the pretrial. That denominator comes out to a standard error of 0.11. And so it comes out to a t of 3.47. Likewise, when we compare baseline to post, we get a t of 3.07. And finally, we want to compare the pre to the post and see if these are different. And we find that no, they are not different from each other with a t of 0.40. So then just a final little summary paragraph here. It says, well, okay, well, the results support hypothesis one. Although all participants completed the trial at the same velocity, participants who had previously established a faster velocity referent experienced greater frustration relative to individuals for whom velocity matched expectations. Furthermore, momentary frustration increased from baseline for these participants. However, measurements taken immediately before and after the trial didn't differ from each other, meaning that a composite of these variables provides the best estimate of frustration during the trial. So we use this composite later on to test subsequent hypotheses that I'm not going to talk about in this video. So I present this as sort of an example. You guys ask me a lot, well, how do I write this up? And I say, well, that's hard for me to answer. It depends on the specific issue at hand. And so you can see here, I'm writing up this conclusion based on what is relevant to the paper. What we want to know is, did frustration change over time? For whom? In what way? And so that's what I've tried to capture with my choice of probing of the interaction, like how I've probed the interaction and how I've written about it. Okay, well, that will do it for this one.